Hello, everybody. Welcome back to today's class where we're going to go over the basics of IRanges and Granges, which we touched on in the last lecture, going over R functions, where we merged replicate peak files into consensus peaks, or learned how that function would work. And we'll see more on that later. But today we want to take a step back and just go over what are IRanges and Granges. And it sounds pretty simple, but it is really a core engine to most of the things we're going to do. So we want to take some time to do that. So I loaded these packages here. And really what IRanges and Granges are is we've been looking at these ChIP-seq peak files, but it could be anything, RNA-seq, whatever. It's an interval that we're interested that has a start and a stop region, essentially. And IRanges likes to think about a start, an end, and a width. But really, you only need two of them to imply where the other one is. So let's take a quick example of IRanges. I'm just going to go over the high-level concepts. Most of this is translated here. But let's make an IRange. So what is an IRange? Well, an IRange is like a ChIP peak profile. So we have a bunch of start and stops of peaks, and we can actually make one of those ourselves. So let's call the object IR1 and call up IRanges. So we've loaded this package and we are now running the function IRanges. And what it wants as parameters is the start. And so we can print the start of 3 different peaks. Let's say it starts at 1, one starts at 200, one starts at 1000. And the first peak ends at 30. And the second peak ends at 299, and the third peak ends at 1200. So this could be space in the genome or anywhere, and any type of data, really. But you can start to see the importance of knowing where the start and stop of something is in the last video where we tried to intersect 2 different things. And that's what we're going to show you more of today here. So what does an IRange look like? Well, let's create this one where we've just made the start and end. We did not make a width. We could have, but it'll do it for us essentially. So let's run that and let's look and see what we got. And so we can see we have an IRanges object and they're all integer values. And here's the start, stop, and width, start, end, and width. And so we can also just grab the start if we wanted with the start IR. So I'll just give us that first column. And basically, you can just call up each column individually with end and width. Plot ranges will allow you to look at ranges. This is really only used when you have a small number you're zooming in on. And what plot ranges is actually a function that we have in our util plotting R functions that we loaded. And if we look down here, we can see there's this function plot ranges. This is the nice thing about R is you can Google what you're trying to do is plot IRanges, and there's a website with a plot ranges function. We went over functions. Here's the parameters it's looking for. Here's its syntax. But now we can just call plot ranges because we have it in our util folder. So that's kind of handy. That's in there already for you as well. So let's plot what does this look like? It looks pretty much like we looked like in the IGV and UCSC visualization. You can see sort of the start and stop of, let's say, a peak in our case right now. Okay, so there's 3 peaks. Well, let's make a replicate peak file and say, here's where they start. And we did this on purpose, so 2 of the 3 will overlap. You can go through the numbers here. But let's just do that. Let's make IR and take a look at IR2. Same syntax we just saw before, start, stop. Now we'll do plot ranges, IR2. And we see it's a little different. There's some overlap, but we might want to see them next to each other. And a nice thing about IRanges is you can simply concatenate them with C. So let's make a new IRange 3, which is the concatenation of IRange 1 and IRange 2, and see what happens there. Okay. So we mixed these 2. And now what we can see is these 2 peaks are going to overlap here. These 2 peaks are going to overlap, but this peak is not going to overlap. And that's important to think about in ranges. It's a little confusing in some of the functions we're going to do when you try and merge these 2 things. There's some considerations more to think about, But we can already see with ranges, if we think of this as a ChIP-seq data set with 3 peaks, that we see that 2 of them are going to overlap and 1 of them won't already here. Okay, so what we can see here with IR3 is that we don't really have anything in this column here that indicates what this file is, like what we're looking at. And it's good to have a name there. And so the names base our function, we can put names into IR1 by simply pasting in what we want to put the name to be, let's call it replicate 1. And we want to tell the slots to put it in. So 1 through 3, because we have 3 here. You could do I through length if you wanted to for a longer vector. And then we'll separate it by a space. So this is just the commands of paste. They're going to be now put into names and placed onto this ranges. So we can name IR1. And so when we look at IR1 now, we can see, oh, this has a little more meaning to us than 1, 2, 3. We could call it peak 1, 2, 3, or intron 1, 2, 3, exon 1, 2, 3, anything we're referring to. So let's name the second IR names. Replicate 2, same thing. We don't really need to look at that. Okay, so overlaps are where we really want to get to. So we're going to make consensus peaks, which we did in going over the make consensus peak function, where we merged 2 long genomic ranges of peaks from 2 different ChIP files and into 1 consensus peak, or where all the peaks overlap. But we're still taking a step back into IRanges before we move into GRanges. And just look at ways we can do overlaps within 1 string of ranges or list of ranges. And so union will sort of merge 2 IRanges. So let's see what the overlap of IR1 and IR2 looks like. If we do that, and we do plot ranges for OV1, what we can see is something what we might not have expected. So we didn't intersect these. What we did is merge them. And what we can see if we look at OV1 here, and what we got is this second peak didn't overlap, but it now has a range from 100 to 299. And that's because when we set it here, the lowest number for peak 2 was 100, and the highest number for peak 2 in the 2 list was 299. So it just made a new range from 100 to 299. It simply collapsed those 3 values. Okay, so that is the union, which you might think of as like, oh, I found my overlaps in the peaks, but that's not quite how IRanges thinks of it. Okay, so that's just to show that in IRanges, that union is going to merge 2 ranges together. And that could be useful if it was exons. Let's say you had multiple exons and you just wanted to merge it into the biggest exon. That would be a good use of union. But really what we're thinking in this consensus peak things is what peaks overlap, and then if they overlap, where do they sort of intersect? And that's shown here with, let's make OV3, where we intersect these 2, and then plot that. And that's more like what we thought we had seen, where we have this range has a little bit of overlap, and you can go back up and look at the numbers, but these are where they actually have overlapping values. So if you wanted to find the minimal intersection, that would be here, or you could find union. And in find overlaps, we'll do something a little different where we can set how much overlap we want there to be. But in IRanges is still pretty powerful that you can just simply merge or find overlaps or intersect them, essentially doing that. So let's actually use find overlaps function. Okay, so here we're going to make OV4, which we're going to do find overlaps like we did with union and intersect above, but we're going to do find overlaps. So now we have OV4. Oh no, that won't plot, because let's take a look at OV4. What OV4 did, or what find overlaps did, is it said, okay, the query file or replicate 1 had an overlap with its first index with the first index of the query or the subject. And same with the third one, whereas the second peak did not overlap. So this is really handy information because we can compress where there were and were not overlaps and do other functions to that, as we'll see later. But for now, if you wanted to sort of count the overlaps, then you could see how many numbers there were here, and you would already know how many overlaps you have, and you could count how many were in, how many peaks were in each range as well. Okay, so that is great, but what the problem is with IRanges is in the human genome, we have multiple chromosomes. So what if the start stop were the same, but they were on different chromosomes? That would be a big problem. So GRanges was developed to take care of that, in which we have certain levels that are remembered by in the IRanges. And what you can see right away here, there's a little more explanation above, but right away we can see we're using ranges in GRanges. We're using IRanges, which we just sort of walked through. So it's really similar to making an IRange, but with GRanges, there's a few things to remember, and they're sort of described below. But seq names is going to tell you what the chromosome is. So we need to really think about chromosome. The strand of the chromosome is important, because if we do a function to this GRanges, if it's on the minus strand, we might want to subtract, versus if it's on the positive strand, we might want to add numbers, and GRanges can take care of that. And then we want to know the ranges as well. So we simply set those with seq names and concatenate. You could make a list of all the chromosomes if you wanted. The strand you could have on the plus strand, the minus strand of DNA, or you don't know, and it would sort of ignore it. And then the last parameter here is ranges, where we call up IRanges to set a new set of starts and stops of 3 different peaks. So we're going to do the same kind of exercise where we pretend there's 3 peaks in 2 different ChIP replicates. Okay, so we'll make this GR here. And now let's take a look at this, and it's going to look a little different than what we just saw before. So it's saying it's a GRanges object, so that's different than an IRanges. And we have seq names, ranges, and strand, which we just put in here. Okay, so that's great. So now we can know what the chromosome is and where the start and stop of a feature is, like let's say a promoter or an exon would have strandedness, whereas maybe a DNA binding element wouldn't matter what strand was implied. So, and we set the ranges to where the peaks are. Okay, so that's great. And I think seq info is a good function to learn from genomic ranges. And again, please explore the manual package more. We're just going through the essentials for what we'll need, but let's look at the seq info GR here. So what it's saying for seq info is it only has one thing, seq names, which is chromosome X. It doesn't have seq lengths, that's missing. Is circular, so is your genome circular? It wants to know. That's important in coordinate space if it could loop back around, and what the genome version is you're using. So GRanges wants to keep track of these things because it knows it's really important. Some of them are not, some are. Like you want to know the length of chromosome X, for instance. You want to know if chromosome X is circular, and you want to know what genome that chromosome is from. So we can simply add these things now that we know what they are, and say, okay, let's add the seq lengths of GR to be set to chromosome X is 1,000,000 base pairs. So let's run that. And now what we can see is we've told GRanges the length of the total chromosome. So let's say you have these 3 peaks and you want to know what percentage of the chromosome they covered. You could now just divide by seq lengths. Okay, so seq info, seq lengths, seq levels is another one. Let's take a look at that. And so we only have one seq level, and that is chromosome X, and seq lengths we will now have here because we just added it above. Okay, so we can add more seq levels, and these are really important because imagine in each ChIP peak file, we have peaks, but we also have what chromosome they're on and where their start and their stop is from the bed format we talked about in UCSC video. And so having the chromosome is really important, and then we can intersect chromosome by chromosome rather than trying to merge complete lists of starts and stops like we did above. So, but we can add more of these levels quickly here. And just now we'll see that we have chromosome Y added to GR. Now, if we look at the seq levels of GR, we can see that there's 2 entries, chromosome X and chromosome Y. And finally, we don't really need to add any of these chromosomes as circular, but we could add the genome version that we're on and say that the genome that we're calling up is going to be HG38 or human genome version 38 here. And now when we look at that, what we'll see is that 2 sequences from HG38 genome. So it knows now the genome that it's on when we look at it. So for now, those are just the very fundamentals of what IRanges and GRanges are. And it's really pretty simple. You'll have to start to learn some of these different commands, but we'll use them so many times they'll become second nature, hopefully. But really just to take home, the take home message here is we're going to have large lists of starts and stops of thousands of ChIP peaks for each factor. And we're going to want to merge those. We're going to want to know how much space they take. The nice thing about knowing the start and the stop is if you sum all of that, which you can easily do in R, then you can know the total space of the genome covered. And we're going to go through all that kind of stuff in the next video where we load lots of GRanges, or what you would call a list of GRanges of features of the human genome, such as promoters, exons, introns, what types of genes. And so we're going to load in annotations of the human genome as GRanges in the next video. But we wanted to give you the basic kind of fundamentals of what we're looking at and what the sort of syntax is. But again, this will become more like second nature as we keep going in the next video and beyond. So thank you very much. And in the meantime, code light.